Hello everyone, and today's topic is the DNA replications, copying genetic information for transmission to the next generation. Occurs in a S phase of cell cycle, and the process of DNA duplicating itself begins with the unwinding of the double helical to expose the bases in the each strand of DNA. Each unpaired nucleotide will attack a complementary nucleotide from the medium, will form base pairing via hydrogen bonding. Enzymes link the alkyl nucleotide by phosphodiester bonds to form a continuous strand. First question asked was whether the duplication was semi-conservative or conservative. Millison and Staff experiment state that semi-conservative, that one strand from parents in each new strand. Conservative means both strand from parents and other it's all new strand. Complementary base pairing produces semi-conservative replication, that double helix. The double helix unwinding each strand act as a template, and complementary base pairing ensures that T signals addition of A on new strand and G signals addition on C. Two daughter helix produced after replication. Overview of DNA replications are: original double helix stand separate, complementary base align opposite templates, and enzyme link sugar phosphate elements of aligned nucleotide into continuous new strand. Experimental proof of semi-conservative replication: three possible models in that: semi-conservative replication, that Watson and Crick model; conservative replication, in that the parallel double helix remain intact, both strands of the daughter double helix are newly synthesized; and dispersive replication, in that at completion both strands of the both double helix contain both original and newly synthesized materials. Next is the Millison Staff experiment confirm semi-conservative replications. Experiment allow differentiation of parallel or parental and newly formed DNA. Bacteria were grown in a media containing either normal isotope or nitrogen or the heavy isotopes, that is N15. DNA banded after equilibrium density gradient centrifugation at position which matched the density of the DNA. Heavy DNA was at higher density than normal DNA. And when bacteria grown in N15 were transferred into N14 containing medium, the newly synthesized DNA strand has a N14 while the parental strand has a N15. They check the composition of the resulting DNA molecules by density gradient centrifugation, found an intermediate bands indicating a hybrid molecule containing both N14 and N15 DNA. Next is what is the mechanism of DNA replications, in that tightly controlled process occurs at a specific times during the cell cycle, and it requires a set of proteins and enzymes and requires energy in the form of ATP, that is adenosine triphosphate. There are two basic steps, that is initiation and elongation. Two basic components in the templates and primer. DNA polymerase, the enzyme that extends the primer, that is pole 3, produces the new strand of complementary DNA, and pole 1 fills the gap between newly synthesized Okazaki segment. Additional enzymes and proteins, that is DNA helix, that is unwind double helix, single stranded binding proteins, that is keep helix open, primase, that is creates RNA primer to initiate synthesis, ligase, that welds the together Okazaki fragments. Next is the origin of replication. Replication proceeds in both directions, that is bidirectionally, from single origin of replication on the prokaryotic cellular chromosome. Replication proceeds in both directions from hundreds to thousands of origins of replication on each of the linear eukaryotic chromosome. What are the origin of replication? In that bacteria have one origin of replication per one chromosome. They only have one chromosome, that is one original. Replication initiation in that DNA origin of replication initiate a protein binding requires DNA helicase opening of DNA strand. Replication initiation is the primase and RNA primer. Replication elongation that DNA pole 3 must have 3 to add 2. Replication is finished. DNA pole remover primer fills gap between 3 ends. DNA ligase connect frag uses 5 ends. So this is all about replication termination in that the ends of the chromosomes cannot be replicated on the lagging strand because there is no primer available. Telomerase enzyme that contain RNA primer which extend the ends of chromosome not normally expresses in significant level. Telomers telomers form a sort of single stranded cap around the chromosome end to protect them from being degraded. Chromosome ends are progressively shortened with each round of the replication. Old cells with shortened telomeres undergo apoptosis, that is the protective for normal cells, and kill the old and possibly mutate. So this is all about the DNA replications.